HESI A2 Biology Exam Review. Question 1. Seedlings are grown in matched trays under carefully controlled conditions. In cells observed during a greenhouse seedling study, each chromosome is copied to form sister chromatids. Which cell-cycle stage is being described? Option A, G2 phase, Option B, cytokinesis, Option C, S phase, Option D, anaphase. The correct answer is Option C, S phase. Chromosomal DNA is duplicated during the synthesis portion of interphase. Each chromosome then consists of two sister chromatids joined at a centromere. Cell division occurs later after the copied material has been checked and organized. The evidence from a greenhouse seedling study applies this principle in a concrete biological setting. Question 2. Seedlings are grown in matched trays under carefully controlled conditions. During a greenhouse seedling study, a root hair cell loses its ability to carry out polypeptide assembly. Which cellular structure is most directly affected? Option A, lysosome, Option B, mitochondrion, Option C, ribosome, Option D, Golgi apparatus. The correct answer is Option C, ribosome. Amino acids are joined in the sequence specified by messenger RNA. The described failure therefore points to the compartment specialized for that work. Other organelles may support the cell, but they do not perform this immediate task. The evidence from a greenhouse seedling study applies this principle in a concrete biological setting. Question 3. Seedlings are grown in matched trays under carefully controlled conditions. Two heterozygous organisms in a greenhouse seedling study are crossed for wing shape, with T dominant to T. Which probability describes an offspring with the recessive phenotype? Option A, 0%, Option B, 50%, Option C, 75%, Option D, 25%. The correct answer is Option D, 25%. Each parent can contribute either allele with equal probability. Combining the gametes produces one homozygous recessive outcome among four equally likely genotype combinations. Only that genotype expresses the recessive phenotype under complete dominance. The evidence from a greenhouse seedling study applies this principle in a concrete biological setting. Stressed about your HESI A2 exam? We can take HESI A2 exam for you remotely and undetected. We always guarantee a pass and payments only after results. To send us an email, scan the code or tap the link in the description and let's get it done. Question 4. Seedlings are grown in matched trays under carefully controlled conditions. During a greenhouse seedling study, cells synthesize a polysaccharide. Which smaller unit is repeatedly joined to build this molecule? Option A, nucleotide, Option B, amino acid, Option C, monosaccharide, Option D, glycerol only. The correct answer is Option C, monosaccharide. Complex carbohydrates are built by linking simple sugars. The arrangement and type of sugar units determine storage or structural properties. Amino acids form polypeptides, while nucleotides form genetic polymers. The evidence from a greenhouse seedling study applies this principle in a concrete biological setting. Question 5. Seedlings are grown in matched trays under carefully controlled conditions. A researcher conducting a greenhouse seedling study changes salinity and records enzyme activity each day. Which factor is the dependent variable? Option A, enzyme activity, Option B, salinity, Option C, the hypothesis wording, Option D, the researcher's name. The correct answer is Option A, enzyme activity. The dependent variable is the outcome measured in response to an experimental change. Its values are compared across treatment levels or groups. The manipulated factor is the independent variable rather than the measured response. The evidence from a greenhouse seedling study applies this principle in a concrete biological setting. Question 6. Seedlings are grown in matched trays under carefully controlled conditions. In a greenhouse seedling study, a researcher changes salinity and measures enzyme activity. Which factor is the independent variable? Option A, the final conclusion, Option B, enzyme activity, Option C, salinity, Option D, the number written on the data table. The correct answer is Option C, salinity. The independent variable is deliberately manipulated to test its effect. The measured response is the dependent variable. A clear experiment changes one main factor while keeping other relevant conditions as constant as possible. The evidence from a greenhouse seedling study applies this principle in a concrete biological setting. Question 7. Seedlings are grown in matched trays under carefully controlled conditions. Consider the food chain algae, zooplankton, small fish, heron in a greenhouse seedling study. Which organism occupies the primary consumer level? Option A, zooplankton, Option B, small fish, Option C, algae, Option D, heron. The correct answer is Option A, zooplankton. Primary consumers obtain energy by feeding directly on producers. The next consumer eats the primary consumer, and higher levels continue the transfer. Identifying the feeding relationship is more useful than relying only on an organism's size. The evidence from a greenhouse seedling study applies this principle in a concrete biological setting. Question 8. Seedlings are grown in matched trays under carefully controlled conditions. A cell in a greenhouse seedling study contains 9% non-penetrating solute and is placed in a solution containing 5% non-penetrating solute. Which outcome is most likely? Option A. The concentrations prevent all molecular motion. Option B. Solute enters by osmosis and lowers cell volume. Option C. Water leaves the cell, causing it to shrink. Option D. Water enters the cell, causing it to swell. The correct answer is option D. Water enters the cell, causing it to swell. A higher solute concentration inside the cell creates a net movement of water inward through the selectively permeable membrane. The added water increases cell volume. Solute movement is not what defines this water-balancing process. The evidence from a greenhouse seedling study applies this principle in a concrete biological setting. Are you looking for HESI A2 exam actual questions? Visit our website pokerexams.com to access HESI A2 exam study guide with actual questions and answers. Scan the QR code or click the link in the description to get HESI A2 exam answer key today. Question 9. Seedlings are grown in matched trays under carefully controlled conditions. One diploid cell from a greenhouse seedling study completes meiosis without error. Which outcome is expected? Option A. One diploid cell with twice as many chromosomes. Option B. For haploid cells with genetic differences. Option C. Two identical diploid cells. Option D. For genetically identical diploid cells. The correct answer is option B. For haploid cells with genetic differences. Two successive nuclear divisions follow one round of DNA replication. Homologous chromosomes separate first, and sister chromatids separate in the second division. Crossing over and independent assortment contribute to variation among the resulting haploid cells. The evidence from a greenhouse seedling study applies this principle in a concrete biological setting. Question 10. Seedlings are grown in matched trays under carefully controlled conditions. A dividing cell in a greenhouse seedling study shows that chromosomes align at the cell equator. Which mitotic stage is shown? Option A. Metaphase. Option B. Telophase. Option C. Prophase. Option D. Anaphase. The correct answer is option A. Metaphase. Spindle attachments position duplicated chromosomes along the middle of the cell. This arrangement allows sister chromatids to be pulled toward opposite poles accurately. Chromosome condensation begins earlier, and nuclear re- formation occurs later. The evidence from a greenhouse seedling study applies this principle in a concrete biological setting. Question 11. Seedlings are grown in matched trays under carefully controlled conditions. Which observation from a greenhouse seedling study is the best example of an adaptation rather than a short-term acclimation? Option A. A plant wilts during one hot afternoon. Option B. An individual temporarily breathes faster at high altitude. Option C. An animal learns a route during its lifetime. Option D. A heritable feature has become common because it improved reproduction across generations. The correct answer is option D. A heritable feature has become common because it improved reproduction across generations. An evolutionary adaptation is an inherited population-level feature shaped over generations. Temporary physiological adjustments and learned behaviors can help an individual respond, but they are not inherited genetic changes. The distinction depends on heritability and generational change. The evidence from a greenhouse seedling study applies this principle in a concrete biological setting. Question 12. Seedlings are grown in matched trays under carefully controlled conditions. In a greenhouse seedling study, a microscopic cell is observed to contain a circular DNA in a nucleoid region. Which broad cell type best matches this observation? Option A. Fungal cell. Option B. Plant cell. Option C. Prokaryotic cell. Option D. Animal cell. The correct answer is option C. Prokaryotic cell. Cells without a membrane-enclosed nucleus keep their chromosome in a nucleoid region. This organization is characteristic of bacteria and archaea. Eukaryotic cells instead package most nuclear DNA inside a membrane-bound nucleus. The evidence from a greenhouse seedling study applies this principle in a concrete biological setting. Question 13. Seedlings are grown in matched trays under carefully controlled conditions. During a greenhouse seedling study, one group receives the experimental treatment and another is kept under normal conditions. Why is the normal group included? Option A. It removes the need for repeated trials. Option B. It increases the treatment dose. Option C. It guarantees the hypothesis is correct. Option D. It provides a baseline for comparison. The correct answer is option D. It provides a baseline for comparison. A comparison group shows what happens without the tested treatment under otherwise similar conditions. Differences between groups can then be associated more confidently with the manipulated factor. A control does not eliminate all uncertainty or replace replication. The evidence from a greenhouse seedling study applies this principle in a concrete biological setting. Question 14. Seedlings are grown in matched trays under carefully controlled conditions. Cells in a greenhouse seedling study join small subunits into a larger biological polymer while releasing water. Which reaction type is occurring? Option A, ionization. Option B, dehydration synthesis. Option C, hydrolysis. Option D, neutralization only. The correct answer is option B, dehydration synthesis. Building a polymer commonly removes a hydrogen from one subunit and a hydroxyl group from another. The removed parts form water while a new covalent bond links the subunits. The reverse reaction uses water to split the bond. The evidence from a greenhouse seedling study applies this principle in a concrete biological setting. Question 15. Seedlings are grown in matched trays under carefully controlled conditions. A researcher examining a greenhouse seedling study identifies a molecule whose main role is catalysis and structural support. Which class best matches that role? Option A, lipids. Option B, proteins. Option C, DNA only. Option D, water. The correct answer is option B, proteins. Chains of amino acids fold into shapes that allow them to act as enzymes, receptors, transporters, and structural fibers. Function depends strongly on 3-dimensional shape. Water and storage lipids do not provide this range of specific catalytic roles. The evidence from a greenhouse seedling study applies this principle in a concrete biological setting. Question 16. Seedlings are grown in matched trays under carefully controlled conditions. During meiosis in a greenhouse seedling study, homologous chromosomes exchange matching DNA segments. Which effect can this event have on offspring? Option A. It always doubles chromosome number. Option B. It prevents all genetic variation. Option C. It converts RNA into DNA in every cell. Option D. It creates new combinations of alleles on chromosomes. The correct answer is option D. It creates new combinations of alleles on chromosomes. Exchange between homologous chromatids produces chromosomes containing maternal and paternal segments in new arrangements. Gametes can therefore carry allele combinations not found together on either original homologue. The process increases variation without normally changing chromosome number. The evidence from a greenhouse seedling study applies this principle in a concrete biological setting. Question 17. Seedlings are grown in matched trays under carefully controlled conditions. A heterozygous organism from a greenhouse seedling study is crossed with a homozygous recessive organism for wing shape. What fraction of the offspring is expected to show the dominant phenotype? Option A, 1. Option B, 1/2. Option C, 1/4. Option D is 0. The correct answer is option B, 1/2. The heterozygous parent produces dominant and recessive gametes in equal proportions, while the recessive parent contributes only the recessive allele. Half the combinations are heterozygous and express the dominant trait. The remaining combinations are homozygous recessive. The evidence from a greenhouse seedling study applies this principle in a concrete biological setting. Question 18. Seedlings are grown in matched trays under carefully controlled conditions. A diploid organism studied in a greenhouse seedling study has 10 chromosomes in a somatic cell. How many chromosomes should a normal gamete contain? Option A, 10. Option B, 5. Option C, 4. Option D, 20. The correct answer is option B, 5. Meiosis reduces chromosome number by half, so fertilization can restore the diploid condition. Each gamete receives one member of every homologous pair. The calculation uses the haploid number rather than the number of chromatids present during replication. The evidence from a greenhouse seedling study applies this principle in a concrete biological setting. Stressed about your HESI A2 exam? We can take HESI A2 exam for you remotely and undetected. We always guarantee a pass and payments only after results. To send us an email, scan the code or tap the link in the description and let's get it done. Question 19. Seedlings are grown in matched trays under carefully controlled conditions. In a greenhouse seedling study, allele T is dominant for a wing shape. Which genotype is homozygous dominant? Option A, TT. Option B, T. Option C, TT. Option D, TT. The correct answer is option A, TT. A homozygous genotype contains two copies of the same allele. When both copies are the dominant form, the dominant phenotype is expressed and no recessive allele is present. A heterozygote contains one of each allele. The evidence from a greenhouse seedling study applies this principle in a concrete biological setting. Question 20. Seedlings are grown in matched trays under carefully controlled conditions. A specimen from a greenhouse seedling study has a centrioles and no cell wall. Which cell type is most consistent with those traits? Option A, animal cell. Option B, plant cell. Option C, bacterial cell. Option D, fungal cell. The correct answer is option A, animal cell. The absence of a cell wall permits a flexible outer boundary, while centrioles commonly help organize cell division in the relevant biological features. Plant and fungal cells possess walls. Bacterial cells do not contain the same membrane-bound internal organization. The evidence from a greenhouse seedling study applies this principle in a concrete biological setting. Question 21. Seedlings are grown in matched trays under carefully controlled conditions. Organisms in a greenhouse seedling study need a rapidly available source of chemical energy. Which class of biological molecule is most directly used for this purpose? Option A, mineral salts. Option B, steroid hormones. Option C, nucleic acids. Option D, carbohydrates. The correct answer is option D, carbohydrates. Sugars can be broken down readily during cellular respiration to release energy for ADP production. They also serve in short-term storage and structural roles in some organisms. Genetic polymers and inorganic ions do not serve as the usual immediate fuel reserve. The evidence from a greenhouse seedling study applies this principle in a concrete biological setting. Question 22. Seedlings are grown in matched trays under carefully controlled conditions. In a greenhouse seedling study, individuals with an inherited trait survive drought more often and produce more offspring. Over many generations, what is the most likely population-level result? Option A. The allele associated with the trait becomes more common. Option B. The population immediately becomes a different species. Option C. The environment directly writes the same mutation into all organisms. Option D. Every individual develops the trait by need. The correct answer is option A. The allele associated with the trait becomes more common. Heritable variation causes individuals to differ in survival and reproduction under environmental pressure. Those with an advantageous trait contribute more copies of the associated allele to later generations. The change occurs in the population's allele frequencies rather than through purposeful modification by individuals. The evidence from a greenhouse seedling study applies this principle in a concrete biological setting. Question 23. Seedlings are grown in matched trays under carefully controlled conditions. Which source creates new heritable variation in a population studied during a greenhouse seedling study? Option A, mutation in DNA that enters the reproductive lineage. Option B, temporary change in body temperature. Option C, identical mitotic copying without error. Option D, use of a trait during an individual's lifetime. The correct answer is option A, mutation in DNA that enters the reproductive lineage. A change in genetic sequence can create a new allele when it occurs in cells that contribute to offspring. Recombination rearranges existing alleles, while mutation can introduce a novel version. Temporary physiological changes are not transmitted as DNA sequence changes. The evidence from a greenhouse seedling study applies this principle in a concrete biological setting. Question 24. Seedlings are grown in matched trays under carefully controlled conditions. An organism in a greenhouse seedling study displays a recessive wing shape under complete dominance. Which genotype must it have? Option A, TT. Option B, TT. Option C, TTT. Option D, TT. The correct answer is option B, TT. A dominant allele masks the recessive allele in a heterozygote. Expression of the recessive phenotype therefore requires two recessive copies. The observable trait alone cannot distinguish the two dominant -phenotype genotypes. The evidence from a greenhouse seedling study applies this principle in a concrete biological setting. Question 25. Seedlings are grown in matched trays under carefully controlled conditions. A membrane in a greenhouse seedling study separates 89 freely permeable particles on one side from 11 on the other. Before equilibrium, which direction describes the net particle movement? Option A, equally in one direction until all particles stop moving. Option B, from the lower minus concentration side toward the higher minus concentration side using ADP. Option C, from the higher minus concentration side toward the lower minus concentration side. Option D, only toward the side containing larger molecules. The correct answer is option C, from the higher minus concentration side toward the lower minus concentration side. Random molecular motion produces a net flux down a concentration gradient. Movement continues in both directions, but more particles leave the crowded side than return to it. At equilibrium, motion continues even though net transfer becomes zero. The evidence from a greenhouse seedling study applies this principle in a concrete biological setting.